Hey guys, and welcome back to Biologenius, sponsored by Cooper Baum. Today, we're learning about iPSCs, bioartificial organs, and chimeral embryos. iPSCs, or induced pluripotent stem cells, are reprogrammed adult somatic cells into stem cells. Basically, we take some skin cells or some blood cells from adult humans and reprogram them into stem cells that can be used anywhere in the body. Now, they're much better than embryonic stem cells because they can be made more patient-specific, and they're better than lentoviruses or retroviruses because they have a lower chance of being rejected by the host. After we get these stem cells, we can put them in the liver, or the brain, or the heart, and it's all great.
Dude, what are you doing in here?
Bioartificial organs are a mix between building an organ and being donated one. What we do is we can either build or 3D print part of an organ, and then we can mix it with donated cells or grown ones. Or we can make our own. Now, bioartificial organs, when we build a scaffolding or part of an organ, we mix it with living cells. We can replace livers, hearts, kidneys, or injured neurons in the brain. Chimeral embryos are super cool. Basically, the way they work is we take an animal embryo, right? Like a pig. When a pig is going to be born, we will take out some of the DNA in the pig. And what we will do is we will inject a human stem cell and engineer it such that when that pig is growing up, he will grow maybe a liver or a kidney inside of that pig. Now, there are some moral uncertainties about growing human organs in pigs. And to that, I say, sponsored by Cooper Baum. For all those who are still here, let's dive a little bit deeper. Induced pluripotent stem cells are created when somatic cells are taken from an adult and the DNA is reprogrammed such that it acts like an embryonic stem cell. However, embryonic stem cells can be a little bit controversial because you're taking cells from an embryo, and so induced pluripotent stem cells are a great alternative. They can also be tailored more to fit a patient than an embryonic stem cell, so there is a lower chance that the host rejects it. According to the National Library of Medicine, the first case of creating induced pluripotent stem cells was in 2006 at Kyoto University in Japan. They used mouse fibroblasts to create induced pluripotent stem cells, and they worked great. Ever since then, embryonic stem cells have been used less and less, and iPSCs have been used more and more. Now, let's explore some of the challenges and future directions of regenerative medicine and tissue engineering. One major challenge in regenerative medicine is ensuring the safety and stability of induced pluripotent stem cells, iPSCs. While iPSCs offer a patient-specific approach with reduced risk of immune rejection, they also pose risks such as uncontrolled growth and potential tumor formation. Scientists are continuously working on refining techniques to improve efficiency and reduce these risks. Another challenge is vascularization, the process of creating functional blood vessel networks in bioengineered tissues and organs. Without proper blood supply, engineered tissues struggle to survive and integrate into the body. To address this, researchers are using growth factors, 3D bioprinting of blood vessels, and electrical or magnetic stimulation to guide cells in forming networks similar to natural blood vessels. In addition to vascularization, another obstacle is scalability. While lab-grown tissues have shown promise in research settings, producing large, fully functional organs for clinical use remains difficult. Ensuring these bioengineered structures can survive, function properly, and integrate into the body over the long term is an ongoing area of study. Looking ahead, the future of regenerative medicine is promising. Scientists are developing more advanced bioreactors that better mimic the human body, helping tissues grow in conditions that closely resemble their natural environment. Additionally, artificial intelligence and machine learning are being used to optimize the design and manufacturing of bioengineered organs, making the process more efficient and precise. One of the most exciting prospects is the potential for full organ regeneration. While we have successfully grown simple tissues and small organ structures, creating complex, fully functional organs like hearts, kidneys, or lungs remains a challenge. However, with continued advancements in stem cell research, 3D bioprinting, and biomaterials, we are moving closer to a future where custom-built organs could become a reality, eliminating the need for long transplant wait lists. Furthermore, researchers are also exploring the use of gene editing tools like CRISPR to enhance regenerative medicine. By modifying DNA sequences, scientists can potentially correct genetic disorders before cells are used in therapy, making treatments even more effective and personalized. Finally, ethical considerations will play a key role in shaping the future of regenerative medicine. As we continue to explore chimeric research and bioengineered organs, ongoing discussions will be necessary to ensure responsible development and application of these technologies. Regenerative medicine and tissue engineering are transforming healthcare, offering hope for millions of patients worldwide. With continued innovation, we may one day see a world where damaged tissues and organs can be fully repaired or replaced, improving lives like never before. Thanks for watching the last and only episode of Biology. It's on account of it's not real, or it's fake, and doesn't exist. Sponsored by Cooper Baum.